In today's class and video, we are going to look at how to determine the concentration of our plant pigment. So we'll start our introduction by saying many of the colors associated with plant pigments are due to the presence of pigment molecules such as chlorophyll, carotenoids, anthocyanins, and beta-cyanins. And what we also have to note is that chlorophylls are responsible for the green color in plants. An example is chlorophyll A and chlorophyll B, and carotenoids also contribute to the yellow, orange, and red colors in plant. An example of the carotenoids are beta carotene, that gives the orange or good orange color, lutein and xanthophylls that are responsible for the yellow color, and lycopene, which is responsible for the red color. Anthocyanins also impart the red, purple, and blue hues to various plant parts. An example is the water-soluble pigment, often found in fruits like berries, flowers, and leaves. Beta-cyanins, which are also found in some plants, provide the red and violet colors. An example is the water-soluble nitrogen-containing pigments. And what you have to note is that the intense colors of these pigment molecules make them ideal for spectroscopy studies. And each pigment has a unique visible spectrum that can provide a positive identification. So this is just a diagram showing the various types of plant pigment that we have. So as you can see from the table, this is our plant pigment. And we have the photosynthetic pigments and the protective pigment. So under the photosynthetic pigments, we have the chlorophylls and we also have the carotenoid. And we have chlorophyll A and B as an example of the chlorophyll. And we also have xanthophylls, lutein, and carotenes as example of the carotenoid. And we also have, for the protective pigment, we have the anthocyanins, which are responsible for the red, purple, and blue color that are normally seen in plants. So now let us look at our test protocol to determine the concentration of our plant extract. So first of all, we will have to dilute our plant pigment in 1 in 10 and 1 in 100 dilution using acetone. So we also have the UV spectrophotometer and acetone. So now let's look at the test method. 1. Dilute the plant extract pigment in 1 in 10 and 1 in 100 with acetone. So for us to dilute the 1 in 10 dilution, we pick 1 ml of our plant extract and add it to 9 ml of acetone. And for us to do the 1 in 100 dilution, we pick 1 ml of the 1 in 10 dilution and add it to 9 ml of acetone to prepare the 1 in 100 dilution. So when we are done with that, we zero the spectrophotometer with acetone. We then measure the absorbance of the plant extract at wavelengths from 400 to 700 nanometers at intervals of 30. And one important thing that we have to note is that we are supposed to use a glass or a quartz cuvette over here. Now, if you should also look at our plant extract in our quartz cuvette, so we can say that there are no air bubbles, okay? And I expect you to know why this is important before measuring the absorbance. And these are some questions that I also like you to try your hands on. 1, why is glass or quartz cuvette used in this experiment? And 2, why was acetone used for diluting the plant pigment? So let's see our assumed table of results. This is just an assumed table of results. So this table shows us the wavelength and the absorbance values for our diluted plant pigment. So we have the 1 in 10 and 1 in 100 dilutions, and these are the respective absorbance value for each of the wavelength at intervals of 20 from 400 to 700 nanometer. So if you are to plot our absorbance value, this will be the nature of graph that we are going to get. So this graph is just the absorption spectra for both the 1 in 10 and the 1 in 100 diluted plant extract.